The Curious Mind.
All right, today we're going to talk about something pretty incredible. We are unlocking the code that makes you, well, you. We're diving headfirst into gene theory to figure out the blueprint of life itself.
You know, it's a question we've all asked, right? You look in the mirror and you wonder, where did I get these eyes? Or this hair? Is my smile from my mom? My height from my dad? For like centuries, this was one of the biggest mysteries in all of biology. But it turns out the answer is all wrapped up in something called gene theory.
So what is it? Well, at its heart, gene theory is pretty simple. It's the idea that these tiny little instructions, which we call genes, are the things responsible for passing on traits, like eye color and hair type from parents to their kids. You can think of them as the basic, fundamental building blocks of heredity. Okay, to really get this, we've got to put on our detective hats and go back in time. Because for ages, how traits were actually passed down, well, it was a massive, unsolved case file for biology.
I mean, people had theories, of course. The most popular one was that traits just blended, kind of like mixing red and white paint to get pink. But that just didn't add up, right? It couldn't explain how some traits would skip a whole generation and then just pop up again out of nowhere. The mystery needed a real clue, a big breakthrough.
And that breakthrough? It came from, of all places, a monastery garden. And this is where we meet our first major player, our first great genetic detective, a guy named Gregor Mendel. So while most of the scientific world was looking at the huge big picture, Mendel did the exact opposite. He got obsessed with the tiny details. He spent years meticulously crossbreeding thousands and thousands of pea plants, tracking very specific traits, one generation after another.
And this is where his genius really shines. He wasn't just, looking at the plants, he was tracking things. Super clear, opposing traits with this incredible, painstaking detail. He'd cross a tall plant with a short one, a plant with round seeds with one that had wrinkled seeds. And he counted everything, every single outcome.
And bingo! This is where he hit the jackpot. He proved that the whole blending idea was wrong. Traits don't blend. When he crossed a tall plant and a short one, he didn't get a medium-sized plant, he got tall plants. It meant that traits were being passed on as these little individual packets of information. We now call them genes, but he was the first to see them. Okay, so this led him to this brilliant idea of dominant and recessive genes. Let's look at how this works. It's actually pretty cool. So if the gene for tall, let's call it a big T, is dominant, it always wins. A plant just needs one copy of that big T to be tall. But the gene for dwarf, a little t, is recessive. It's kind of shy. A plant needs two copies of that little t to actually be a dwarf. And just like that, this simple model explained everything. It's how a trait like shortness could just vanish for a generation, and then, boom, reappear later. So based on all this work, he came up with some fundamental rules that we now call Mendel's laws. The first one, the law of segregation, basically just means you get one version of a gene from each parent. They're not stuck together. Then there's the law of independent assortment, which sounds complicated but just means that inheriting, say, the gene for being tall has nothing to do with whether you'll inherit the gene for round seeds. They're independent. And these ideas, they became the absolute foundation for all of modern genetics. So Mendel had cracked a huge part of the case. He found the what, these little units of heredity. But the where and the how, still a total mystery. And as you can see from this little case file, it was going to take almost another 100 years to finally crack the whole thing open. All right, so the next big question for our science detectives was obvious. Where in the heck are these units hiding inside the cell? The search was on. It was like a manhunt for the physical carriers of heredity. And they found them. In the early 1900s, with way better microscopes, scientists finally found the hiding spot. They saw these little thread-like things inside cells called chromosomes. And the amazing part was, these chromosomes behaved exactly like Mendel's units were supposed to. They came in pairs, each parent passed on one to their kid. It was the aha moment. The genes weren't just floating around, they were passengers riding on the chromosomes. Okay, so we've got the what, the genes, we've got the where on the chromosomes. The last piece of the puzzle, was the big one, the final boss, if you will. What are genes actually made of? How do they hold all that information? This, my friends, is the climax of our story. And the answer arrived in 1953. It came from the famous work of James Watson and Francis Crick, who, it's really important to say, were building on the critical research of others, especially Rosalind Franklin. Together, they figured out the actual molecular structure of the chemical that carries our genes, and that chemical is DNA. And man, what a structure it turned out to be. This beautiful, elegant double helix. They realized a gene isn't some mystical thing, it's just a specific section of this long DNA molecule. And maybe the most amazing thing of all, the entire instruction manual for building a living thing is written in a simple 4-letter alphabet. Just A, T, G, and C. That's it. Now that might sound a little abstract, but there's a really simple analogy that makes it all just click. Okay, let's break it down. Think of your entire DNA molecule as a giant recipe book, a huge one. Now, each individual gene is like one single recipe in that book. Let's say the recipe for chocolate cake. That recipe gives the cell the instructions to build a protein. That's the final dish, the cake itself. And your trait, like having brown eyes, is just the result. It's the taste of the dish. It's really that simple and elegant. So there you have it. The historical puzzle is solved. But, okay, why should you care? Why is this more than just a really cool science story? Why does gene theory matter so much to us, right here, right now? Well, because what we know now is that genes do way more than just decide your eye color. They are the master blueprints for building all the proteins that make our bodies run. They influence literally everything from how we process our food to our risk for getting certain diseases. And this really shows you just how powerful that code is. Take a tiny little spelling mistake, what scientists call a mutation, in just one gene, the one for hemoglobin. That one little error completely changes the recipe. The protein that gets built causes red blood cells to change their shape, which leads to sickle cell anemia. It's incredible proof that changing just one single letter in that massive recipe book can change everything. And look, understanding this code has totally changed our world. It's the bedrock of modern medicine, right? It lets us diagnose genetic diseases. It's absolutely critical for forensics, DNA fingerprinting. It's the engine behind biotechnology, helping create brand new medicines. It has even transformed agriculture, letting us develop better, hardier crops. This stuff touches pretty much every part of modern life. So if you're going to remember one thing from all of this, let it be this. Gene theory is the whole story of inheritance. It's the how it works, the where the info is stored, and the how it gets passed on, all rolled into one. So we've done it. We've cracked the code. For the first time in all of human history, we can actually read the book of life. And now, with technology like genetic engineering, we're starting to learn how to edit it. So the big question for all of us isn't just what is the code anymore. It's what are we going to do with this knowledge? What are we going to write in the next chapter?
The Curious Mind.